Okay, so going over meiosis again, and we'll compare it to meiosis real quick, and then we'll talk about what goes on in humans and what happens when things go wrong with these processes. So in meiosis again, you start with a diploid parent cell. You undergo one round of DNA replication. And so here you actually have a cell with a foreign DNA content, right? So at this point, oops, it's a bad arrow. But the cells, you have your pairs of chromosomes and you have your duplicated DNA in the sister chromatids. So you actually have a 4N DNA content. That's how we get to 1N, 1N cells by the end of it. So in prophase one, um, the homologous chromosomes find each other. They undergo recombination, synapsis, and recombination, um, and then form recombinant DNA. In metaphase one, the homologous chromosomes pull away from each other. And well, in metaphase one, the homologous chromosomes line along the metaphase plate. And in anaphase one, the homologous chromosomes pull away from each other. This is followed by a second round of chromosome segregation in which, so here's telophase 2 here, in that second round, the sister chromatids now segregate from each other. Um, and so they align along the metaphase plate, perpendicular actually to the original one, and then the sister chromatids are pulled away from each other. The result here is that you have four daughter cells each with the proper number of chromosomes. So here this is two. So you see chromosome 1 and chromosome 2 are in each of these daughter cells. Uh, and these daughter cells are all haploid. So they have, instead of having a pair of homologous chromosomes, they only have one of each. And they are all genetically different from the parent. And so you see this one has one full red chromosome. This chromosome number 2 here is genetically different from any of the chromosome number 2s in the parent cell. Um, and so that is how meiosis works. You have one round of DNA replication followed by two rounds of chromosome segregation to form four 1N or haploid products. Now, change colors. Let's see what's happening in mitosis. In mitosis, you start with a diploid. You have a round of DNA replication. So you have a 4N DNA content. And then you have alignment of chromosomes along a metaphase plate. In anaphase 1, you have segregation of the sister chromatids, which are identical copies of each other. And then you're left with a 2N products, right? And so mitosis, there is a round of DNA replication. There is one round of chromosome segregation. And the result is two diploid products that are genetically identical to the parent. Oh, and so let me just, in purple here, say these are genetically different than the parent. All right, so those are the differences between those two. This is what meiosis looks like in humans, I guess in most mammals. In males, it goes basically as we described. You have DNA replication, you have your first meiotic division, second meiotic division, and then you have four haploid products from this one parent cell. All right. So each parent cell will produce four sperm. In females, it's a little bit different. You have around DNA replication. You have your first meiotic division here, and actually, um, you have the cells are stuck in prophase one until the menstruation cycle where they mature. And so this happens actually almost at birth or before the birth of the female, in which all of their eggs undergo, ovum undergo that prophase, and then they are frozen until they develop during the menstruation cycle. And so then what happens is that the second meiotic division, one of these will become a mature oocyte, and the other ones, the meiotic division occurs, but these become polar bodies, which are, basically um, defunct cells. And so you only produce one product from meiosis, one mature product from meiosis that's capable of fertilization. Now here, this has a 1N DNA content. This has a 1N DNA content. So when there's a fertilization event, you're left with a 2N product that is ready, that is diploid. Oops. Oh, yeah. So that's oh, this is a comparison of meiosis and mitosis here. We kind of went through this before, the differences between the two and how similar mitosis is to meiosis II, because both of these involve segregation of the sister chromatids. And here looks, I said before, that you should be able to identify just by looking at um, what's going on. You should know immediately what phase of the cell cycle you are, whether it's mitosis or meiosis, so forth. So this is metaphase one, in which the homologous chromosomes are aligning along the metaphase plate. This is anaphase one, in which the homologous chromosomes are segregated. Um, and then telophase one, prophase two, we now have the two cells that are now going to divide their sister chromatids. This is metaphase two. We know that because there are two of them that are in these separate cells. Anaphase two, in which the sister chromatids are segregating from each other, and telophase two. So that is how things should be, but there's lots that go wrong. We call aberrant chromosome segregation and meiosis non-disjunction events, and they occur all the time, but we usually don't see the products of it because these chromosomes are necessary in the proper amount in order to be viable. And so here's a normal meiosis. This is for two, um, for one chromosome. So in humans, we have 23. This is just looking at one, right? As we said before, in normal meiosis, you have four haploid products from one diploid parent cell. Now, if you have a non-disjunction event, you have a failure to segregate in one of the meioses. So a first division non-disjunction event, non-disjunction, failure to separate, it means that the homologous chromosomes fail to segregate. And we can look at the products of meiosis and immediately determine what type of non-disjunction event you have. And so note here, in a meiosis 1 non-disjunction event, then the homologous chromosomes fail to segregate. So you have both homologous chromosomes going to one pole, and then you have another one that doesn't receive that particular chromosome. And if we look for that chromosome, erase here and change the color. what do we see? Well, meiosis II occurs fine. So the sister chromatids segregate, all right? And so you will have two of the cells that will have a diploid chromosome number, and these are genetically different from mom and dad. And so that's in two of the cells, and then you'll have two of the cells that are empty, right? So this is bad. We'll see. This kind of kills the most cells for most chromosomes. We can only tolerate, there are only a handful of chromosomes that can tolerate non-disjunction events. Now, if you have a meiosis II non-disjunction event, second division non-disjunction, then what happens? The homologous chromosomes segregate fine from each other, but the sister chromatids fail to segregate. And so here in this one, sister chromatids segregated fine, but in this one, this sister chromatid failed to segregate. So in a meiosis II non-disjunction event, you have two products that are fine, one product that has a diploid, well, it's not diploid, but has a two times the chromosome for whatever chromosome you're looking at, and one that doesn't have it at all, right? So by looking at the products of the meioses, um, um, you can tell whether you have a first meiosis one non-disjunction event or a meiosis two non-disjunction event. And if you look, I think there's some alignment issues here. Um, oh, no, that's it. This is chromosome 21. That's very confusing. This is kind of what happens with these non-disjunction events. They cause trisomies. So if you have a non-disjunction event, then that means that you'll have one chromosome that will have three or one uh, zygote that'll have three chromosomes for a particular uh, chromosome versus two. And you see in general, if you look here at the other autosomes, right? In general, if there is a non-disjunction event, then you get a spontaneous abortion. Um, and so for most of our chromosomes, we cannot tolerate having an additional chromosome or missing a chromosome. And so those are not viable organisms, and usually they spontaneously abort relatively early. It's thought that up to 30% of successful conceptions, fertilization events, result in spontaneous abortions because of um, uh, aneuploidy, aneuploidy, oops, I think I spelled that wrong, which are abnormal chromosome numbers. Okay, so for most part, uh, we can't tolerate that. There are some chromosomes in which you can. So note trisomy in chromosome 13. Most of those result in spontaneous abortion, but there's a little sliver of blue here that indicates that there are some live births. In chromosome 18, you see the same. Most resort in spontaneous abortion, but there are a couple that occur. And then trisomy 21, you see here, let me just erase this box, that you do get a lot of spontaneous abortions, but there is a significant fraction of live borns. And we should recognize trisomy 21 as Down's syndrome. And so you have successful live births, although there are some consequences to that in terms of morphological differences and mental cognitive issues. And then the other chromosomes that we can tolerate um, having an abnormal number amount of are the sex chromosomes. So, we generally can't tolerate having only one of X chromosome, but you see, we can tolerate an XXY. And you see, when that happens, many of those are live births. We can tolerate XYY, and then we can tolerate XXX as well. And so there are some issues, there are some morphological issues with abnormal sex chromosomes, but in general, these are live births. And then there are other things that happen, translocations, which are chunks of chromosomes that move from one place to another. If they're balanced, meaning that they're cut into place and you don't lose any genetic information, many of those are live born. Unbalanced, that cause a loss of genetic information, um, um, don't don't result don't work out very well. And then we can't tolerate at all polyploidies, which are duplications of the top entire chromosomal um, um, system. And so this is looking at of those live birds, so looking at all the live birds, and we see the vast, well, not vast majority, but the majority are trisomy 21. Um, And then well, I guess this is more the majority here, looking, many of the people live born will be the trisomies of the sex chromosomes. There are some issues. Single X is Turner syndrome, XXY is Klinefelter syndrome, and so forth. And so you see here, trisomy 18 and trisomy 23, 13, you only get a small proportion of the number of live births. And then looking at having extra sex chromosomes here, Klinefelter syndrome, you tend to be taller, feminized physique, poor beard growth, uh, abnormal genitalia development. Most of these, I believe, are not fertile, cannot reproduce. Turner syndrome, short stature, thick neck, heart issues, and underdeveloped sex structures as well. So human, they are um, vi viable, but still have some effects of the non-destruction events. All right, so that wraps up the cell cycle chapter, and we will move to central dogma for probably the rest of the term.